So, we can say the sensitization, it was happening due to the presynaptic facilitation via the serotonin neurotransmitter release that we discussed in the previous section. Now, coming to the next term, that is the long term potentiation, means if there happen the persistent enhancement of the post synaptic potential. The word persistent is key word over here. If persistently enhanced post synaptic potential response to the presynaptic stimulation after a brief period of rapidly repeated stimulation of the presynaptic neuron, that is called as long term potentiation. It thou resembles to the post technique potentiation, but that was lasting for certain time period of like say minutes or hours, but if it lasts for days, then it is long term potentiation. So, what is the basic difference between this long term potentiation and post technique potentiation is, one is it will last for days or weeks. Okay. It is initiated as the PTP, the post technique potentiation, it is initiated by increase in intracellular calcium in either the post synaptic or the presynaptic. What was happening in the post technique potentiation? Only in the presynaptic neuron. So, that is the second difference. The long term potentiation due to increase in intracellular calcium either in the presynaptic or the post synaptic potentiation. And this post long term potentiation, it was studied in greater detail in hippocampus at a certain this pathway that is called as Schaffer collateral. So, what is this Schaffer collateral, that we will discuss in detail in this slide, but before going to the Schaffer collateral, let us discuss about the basic component. There are two type of the receptors. This Schaffer collateral, this is nothing but it is actually kind of a connection between the presynaptic and postsynaptic neuron, and that postsynaptic neuron has got the NMDA receptor. What is NMDA? N-methyl D-aspartate receptor, and the accompanying to the NMDA receptor, like this is the NMDA receptor, there is one more receptor, that is AMPA. A for alpha amino 3 hydroxy, M for 5 methyl isoxazole, and PA means propionic acid. So, there are these two type of the receptor, they are located in the postsynaptic neuron. When the presynaptic neuron, it releases glutamate. This glutamate, it has got the binding site at the NMDA as well as AMPA receptor. Okay. So, it will bind, attach to both of them, but because of the attachment of the glutamate to the AMPA receptor, there is influx of the sodium ions, right, influx of the sodium ion happening. But just because of the attachment of the glutamate at the NMDA receptor, this NMDA receptor won't open up. Why it is not opening up? Because now there is a receptor for the glutamate, but it is blocked by the magnesium channel. If we look at this thing, see, this is the closed NMDA receptor, like it is closed over here, and it has got the site for the attachment of L-glutamate. This is site for attachment of the glutamate receptor, the neurotransmitter that is released, and there is one more site that is for the glycine, and along with those there is a blockage or the binding site for the magnesium over here. So, when the glycine and glutamate, they bind to the receptor, the closed ion channel opens up, but at the resting membrane potential, like when the glutamate and glycine attach, it will open up, but there won't be any flow of ions through this channel because it is blocked at this way with the magnesium ion, and this magnesium ion will get removed only after increase in the membrane potential. When the glutamate and glycine attaches, it brings about the resting membrane potential, all these things is happening. Okay. So, this channel is blocked by the magnesium at the resting membrane potential. This block is removed if the partial depolarization is produced by the other input, right. So, we can say NMDA receptor in itself, if there is NMDA receptor only and glutamate is getting released, it won't open up. Glutamate will come and get attached over here, but the magnesium is blocking this channel. So, there won't be any inflow or outflow of the certain ions through the NMDA receptor. So, it needs the company of other receptors which changes the resting membrane potential, and that other receptor happens to be AMPA at a certain connection pathway of the hippocampus, that is called as Schaffer collateral. We will talk about in detail about. So, NMDA is accompanied by AMPA, glutamate gets attached to NMDA as well as AMPA. First it gets attached to the AMPA, this will open up, there will be influx of the sodium ion. So, the membrane potential will increase from the resting membrane potential, and that increased or slight depolarization that is happening in this postsynaptic neuron will make the magnesium to get removed, it will get unlocked. Now, there will be inflow of the calcium ion. Okay. So, if we look at this Schaffer collateral, what is happening over here, let me show this thing. See, this is the presynaptic neuron and this one is the postsynaptic neuron, and these are glutamate, they are getting released. Okay. Now, this glutamate, what it is doing, it is getting attached to AMPA receptor. This is AMPA receptor, these two are 1 and 2 are the AMPA receptor. It gets attached, then through the AMPA receptor there is influx of the sodium ion and outflux of the potassium ion. Okay. So, because of the opening of this thing, what is happening, the resting membrane potential will become moved more towards the positive potential. Okay. There will be the depolarization, not the maximum depolarization, slight depolarization, and because of that slight depolarization, this magnesium channel, this blockage will be removed. Now, through this NMDA receptor, sodium influx happens and the calcium influx happens, because this is permissible for the sodium as well as the calcium. AMP receptor was not permissible for the calcium. So, this calcium will enter and this calcium will get attached to the calmodulin, calcium calmodulin kinase. This is calcium calmodulin. Calmodulin means cal stands for calcium, modulin means gets modulated because of the presence of the calcium. So, that calcium calmodulin kinase, this calcium and formation of the like this, we can say the complex of the calcium along with the calmodulin, it will activate calcium calmodulin kinase, then this PKC, this is protein kinase C and tyrosine kinase. All these three kinase, calcium calmodulin kinase, protein kinase C and tyrosine kinase, what does it do? They mostly either increase the transmission neurotransmitter via the AMP receptor or bring about more and more AMP receptor from cytoplasm to the lumen. Okay, to the lumen. See, these receptors, they are getting synthesized or generated within the cytoplasm and they are brought to the membrane, plasma membrane. So, this calcium calmodulin kinase first increases the attached AMP receptors conductance. Secondly, it brings about more and more AMP receptor to get attached to the membrane and then process of this sodium influx. So, there will be enhanced depolarization, right. That is one mechanism. The second is this calcium calmodulin kinase along with the protein kinase C and tyrosine kinase. It brings about certain changes in the synthesis machinery, like some protein synthesis machinery. Before we go to that protein synthesis machinery, let me say what is happening because of the increased number of the AMP receptor. There is more and more generation of the excitatory postsynaptic potential within this postsynaptic neuron, right. That is a kind of the post-technic potentiation similar to, right. Now, the second thing is it was happened because of the stimulation. We provided the stimulation at the presynaptic neuron, it released glutamate, and glutamate gets attached to the AMPA, right, to the AMPA, and then there is opening of NMDA receptor, more and more attachment of the AMPA receptor to the surface. So, more and more generation of the excitatory post-synaptic potential and more generation of the action potential in this post-synaptic neuron. Now, the most important feature is these calcium calmodulin kinase, protein kinase C and tyrosine kinase brings about the changes in the synthesis machinery, and this synthesizes a certain signal, and that signal, now we are not certain, but scientists believe that signal happens to be the nitric oxide. It gets released from the postsynaptic neuron, travels and goes to the presynaptic neuron. What does it do? It enhances the glutamate release. See, the triggering point is generation of the action potential within the presynaptic neuron. We provided a stimulus. That stimulus released the glutamate. Now that stimulus has been removed. If that stimulus has been removed, that glutamate release will also get reduced. It will get stopped. So there won't be any generation of the potential in the postsynaptic neuron. But this glutamate, what it did? It did the generation of a certain signal generator, which started a cycle. Because of this generation of this signal that happens to be the nitric oxide, it goes and it helps in releasing of the glutamate. Now there is no need for the stimulus, and there is release of the glutamate. That release of the glutamate will increase more and more calcium, more and more generation of that regulator, and more potentiation. So automatic a cycle happens. So that is called as long term potentiation. Okay, that happens in the hippocampal area. Okay, hippocampal area, and hippocampus is, that's why we say the hippocampus is the part of the temporal lobe, medial temporal lobe, that is needed for the memory storage, for the generation, like the short term memory, conversion of the short term memory to the long term memory. Okay. Now exactly opposite of the long term potentiation is the long term depression. Now long term depression, this kind of process also happens in the hippocampus and at certain areas, certain parts of the cerebellum. So it is opposite of the long term potentiation. It is characterized by decrease in the synaptic strength, totally opposite of the long term potentiation. It is produced by slow stimulation of the presynaptic neuron, and it is associated with a smaller rise in the intercellular calcium than that occurs in the long term potentiation. So what is like the main mechanism of action happening? Due to the phosphorylation of GLU-R2 subunit of AMPA receptor. GLU-R2 is glutamate, like say there is a channel, that channel glutamate receptor, this receptor, it has got certain subunit, multiple subunit. So one of that subunit, that is the GLU-R2 subunit of the AMPA receptor, it gets phosphorylated, and because of the phosphorylation of this certain subunit, there will be decrease in the conductance of the AMPA, right, and decrease in the movement of the AMPA receptor. See, in the long term potentiation, what was happening? There was increase in the conductance of the AMPA receptor and more movement of the AMPA receptor from the cytoplasm to the surface. Now what is happening in this one? Reduced conductance, and the secondly, less movement towards the lumen side. Instead of from the lumen side, it will move toward the cytoplasm. So there will be less generation of the excitatory post-synaptic potential, and there will be reduction in the generation of the action potential. And it happens in certain areas of the cerebellum, like in learning process, certain sort of the learning process in the cerebellum. This type of the long term depression happens over there.